Good afternoon, everybody, and I am very thankful to the Yakult Foundation and the supporters, THSTI and Apollo Hospital for giving me an opportunity to share with you my research experience with my research team. And I am also grateful to the chairperson, Professor Ramakrishna, and Professor Shinjani Bhatnagar to chair this session. And I am going to take you from the dietary habits that has been talked to, fermented food that has been talked to. I am taking you to a different story of comparative assessment of the predominating gut flora during inflammatory bowel disease. Now, as we know that this inflammatory bowel disease is an autoimmune disease, and there is no pathogenic organism has been found so far to be associated with the disease. And it is a multifactorial disease. It has a genetic component, and it also has a microbial component which exaggerates the immune response. So far this is the story that we know about the inflammatory bowel disease, which involves two diseases, ulcerative colitis and Crohn's disease. And the study that we have done in our laboratory mainly concentrates with the ulcerative colitis patients, which we have collected the samples from the gastroenterology department of All India Institute of Medical Science with my collaborator, Dr. Vineet Ahuja. Now, coming to the contributions of the microbiota to the host, which has been deliberated in detail in the morning session as well as in the afternoon session. And what we see that the digestion and the fermentation of the carbohydrates are is very important contribution that the microbiota makes, and it also involves in the production of vitamins, development of the GALT, that is the gut-associated lymphoid tissue, polarization of the gut-specific immune responses, and the prevention and colonization by pathogens. And in turn, the gut immune responses that are induced by commensal populations regulate the composition of the microbiota to maintain the homeostasis. Now, a paper which appeared in Nature Review Immunology in 2013 says that the gut plays a protective as well as a pathogenic role. Now, in a normal individual, we find that the gut microbiota is in a symbiotic relationship. And the types of bacteria that are present so far, of course, the genome projects have come and microbiome projects have come, but still there are many bacteria which still remains unclassified. So there are certain unclassified bacteria, there are certain beneficial bacteria, and there are certain pathobiomes. Now, the purpose of these pathobiomes are that the useful bacteria keep the pathobiomes population down, so that the gall development and the Th17 cells, Th1 cells, which are responsible for the production of the pro and anti-inflammatory cytokines, can take place, and it also helps in the barrier function, Reg3 generations, etc. And it also plays an important role in the production of Treg cells and the anti-inflammatory IL-10 cytokine. And these symbionts also keep, in the due course, they keep the pathobionts in control. But when a dysbiosis takes place, or when there is a disease and there is an alteration in the gut flora, it seems that the dysbiosis, because of the dysbiosis, there could be a production of the pathobionts, which are not coming from outside. They are indigenous flora. But some of these pathobionts might dominate during the disease condition. And because of this, it disrupts the microbial community structure, and there is an accumulation of cholitogenic pathobionts. The pathogenesis of IBD is mainly located in, if you look at the pathogenesis, we find that it is located in the areas with high bacterial concentration. As I told you, there are two diseases. In case of Crohn's disease, it is mostly located in the terminal ileum and cecum, and in case of ulcerative colitis, it's the rectum area. The central event in the pathogenesis of IBD is the exaggerated immune response to the commensal intestinal bacteria or to its specific members. And this idea came because in a genetically modified animal model of IBD, do not develop colitis under germ-free conditions. So this clearly shows that there is a huge role of the microflora or the indigenous microflora in the manifestation of this disease. Now, we started our work somewhere around 2008, and that time we are working on the mucosal flora of the biopsy samples that we took from the hospital. And our results had shown clear delineation of the concentration of the mucosal flora between the predominating and subdominating genera under disease conditions. And in the previous speaker has already pointed out which are the dominant and subdominant flora in the gut, indicating that the subsets of bacteria participating in the pathogenesis of ulcerative colitis and Crohn's disease are likely to be different. And in the present study, what I am going to present today is to show, to evaluate and compare the changes in the diversity of fecal flora in the Indian patients. Right now, we are working on the fecal flora of these patients from healthy to disease state during ulcerative colitis, and also quantified the bacterial population using certain specific probes that we have generated in our lab in control versus patient samples. And in the patient samples, we have taken 3 categories of samples: moderate, severe, and remission stages of the disease. And we tried to obtain any significant association of the bacterial flora during the disease condition. Now, these are the dominant and the subdominant bacteria of the human gut, which we have studied in our samples, that is Bacteroides, Bifidobacteria, Clostridium coccoides members, members of Clostridium leptum, Enterobacteriaceae members, Lactobacillus, Enterococcus, Atopoboium, Methanogens, and sulfate-reducing bacteria. And the techniques that we have used to illustrate these changes or the fluctuations in the gut flora involves fish microscopy, fish flow cytometry, and qPCR analysis. And we have also looked into the metabolites, the changes in the metabolites by GC analysis. And we have tried to do a preliminary study, this is still an incomplete study, to figure out the important changes that have taken place at the species level by using a denaturing page. And the probes that we have used for the flow cytometry are the EUV probe, that is positive control probe, for the to measure the total bacteria which is leveled with FITC, and a non-UV 338, which is to see if there is any background signal, that is with non-UV 338, which should not amplify any of the bacterial species. And the genus specific probes that we have used were leveled with Psi5 at the 5' end. So this is the quantification of the Bacteroides prevetola group. Here we show the fish microscopy of whole cell in situ hybridization of the straight from the fecal sample with genus specific Psi5 leveled probe. So this is for the Bacteroides prevetola group, and down you can see the fish flow data which we have generated. And if you look at the graph, you can see that there is a control, there is a moderate, there is a severe, and the remission cases. And if you see carefully, you will see that there is a relationship when we compare the control, the severe and the remission. We see the fecal flora coming down, both by flow cytometry as well as the qPCR analysis of this Bacteroides flora. And this Bacteroides actually goes significantly high in the severe patients if we compare it with the control individuals. So the population of Bacteroides increases significantly during severe condition and reverts back to normal during remission. Now, when we quantified the genus Bifidobacteria, again with the flow cytometry, we find that there is a significant reduction which is expected in the Bifido population during the disease condition. But what we find that in the remission patients, again the flora comes back towards the normal level. And this was proved both by flow cytometry as well as the quantitative PCR analysis. And on the upper panel, we show the bacteria that are stained with the Psi5 leveled Bifidobacterium probe. So the population of the Bifidobacteria drops significantly during severe condition and reverts back during the remission stage. Coming to the Clostridium coccoides group, again we find that there is a significant drop in the moderate, followed by the severe condition of the disease, and the patients which are in the remission stage again seems to revert back to the normal level of Clostridium cocoides, which is proved again by flow cytometry and the qPCR analysis. The members of Clostridium cocoides drop significantly during moderate and severe condition, but reverts back during the remission stage. In case of Clostridium leptum, it was very interesting that we found that there was a drop of members of Clostridium leptum group, and there is a drop in the moderate as well as in the severe cases. But in the remission cases, we did not find a significant turnover or significant reversion back to the normal level, both by flow cytometry as well as by qPCR. So this shows that the members of the Clostridium leptum group, which actually comes down, they do not revert back so soon. It takes time probably. So the population of Clostridium drops significantly during this disease condition, but do not revert back during remission stage of the disease. So when we saw that there was a significant reduction in the butyrate producing bacteria, that is the members of the Clostridium group, we went little bit in detail to design primers and the probes for Faecalibacterium plasmodium, Roseveria intestinalis, and Eubacterium heli in particular. And we again quantified them using the fish flow and the qPCR analysis, which again showed that the members of Faecalibacterium plasmodium coming down in the severe condition and showing a tendency to come back at the remission stage. And so is with the Roseveria intestinalis. But Eubacterium heli, we did not find a significant change. And this was true both by fish flow and the qPCR analysis. The decreased abundance of predominant butyrate producers like Roseveria intestinalis and Faecalibacterium plasmodium in disease condition was quite remarkable. And then comes the Lactobacillus enterococcus group. Now the members of Lactobacillus enterococcus group, what we find that in case of the moderate conditions as well as in the severe conditions, we find that there is an increase in the lactobacillus, which was a big surprise for us. So we are expecting that the lactobacilli population would come down, but maybe because of the dietary habits of Indian, population, the production of lactobacilli, even in the control samples, were higher when we compared with the literature in the European world. We find that the lactobacilli population actually goes up, and in the remission condition again we find that it is coming down to the normal flora. So this was something which was difficult for us to explain at this point of time. So the increased abundance in members of lactobacillus during disease condition was seen in our study. And these are the subdominant flora known as Atopobium cluster. And here also we find that even in the severe conditions, the population is low, but it goes up. So we don't know if all of them are pathobionts or not, but we find that there is an increase in the population of the Atopobium cluster during the disease condition. So there was a significant increase in the Atopobium during severe condition of UC. And in case of Desulfovibrio, again we find that the population of Desulfovibrio, that is the sulfate producing bacteria, are going high in case of moderate and severe conditions, whereas in case of remission they still maintain the level as we see in the severe condition of the disease. And this was seen both in the flow cytometry and the qPCR analysis. Now coming to the methanogens, this was very interesting that we found an increase in the Desulfovibrio bacteria, but we found a decrease in the methanogens. The population of methanogens of course is low in a normal individual, but what we find that in the moderate and the severe conditions, the methanogens actually come down, and we specifically saw the quantitative analysis by PCR for the Methanobrevibacter smithii, and we find that in case of severe condition, we find a little high in the Methanobrevibacter species, but it was not significantly high. So the p-value was above 0.05. So there was a decrease in the methanogens during severe condition of UC. In this slide, what I show you is we made a comparative study of the firmicutes versus bacteroids the members of the firmicutes versus bacterioids during control, moderate, severe and remission study. What we find that in case of moderate and the severe condition, especially in the severe condition, the ratio of firmicutes to bacterioids come down. So that means the bacterioids population go up, firmicutes go down. And during the remission, they sort of come back, revert back to the normal level. When we made a comparison of the gram-positive versus gram-negative group, what we find that the members of this ratio of gram-positive to gram-negative, that is, there is an increase, there is a decrease in the ratio of gram-positive to gram-negative because there is a change in the qPCR analysis. And in order to establish a probable link between the altered butyrate producing bacteria and the impaired cellular metabolism, we also check the metabolites, that is the short chain fatty acid production, using gas chromatogram profile of the fecal samples during these disease conditions. So what we did was, there is a standard, and we took the fecal samples and measured each of these metabolites. So there are, if we look into this figure, we find that there is a significant drop in the acetate production, in the propionate production, and in the butyrate production when we also measured the butyrate level in the fecal sample itself, which corroborated our observation where we saw that there was a drop in the butyrate producing bacteria. So among SCFA, butyrate and propionate concentration decreased significantly during the disease condition. And there was an increase in the lactate concentration. As I told you that we also found an increase in the lactic acid bacteria. So to prove this, what we did, can I take 2 minutes, please? So what we did was, we did a RICE study where we took a forward primer from the 16S ribosomal rRNA and a reverse primer from the 23S rRNA, and we wanted to amplify the variable region, and we took a genus-specific primer for, say, lactobacillus in this example. I'll just show you one example here, where we have seen that there is a severe condition and the moderate and the control and the remission. We find that if we put it on a DNHN page, we can see the number of bands which are different in these three conditions. So what we did, we took these bands and we sequenced them, and we went back to the database, and what we find that the population in the moderate, severe and remission, the bacteria which it shows matches are quite different. So similarly we did for the bacterioids, we did also for the, I'll just run through these slides, and we find that some of these bacteria are, for example, induces colitis in IL-10 deficient mice, this back one which was common in all category. So some of these bacteria could be connected with their functions as well. We did for the Clostridium leptum as well. Then we also did a phylogenetic tree of Clostridium leptum showing the sequence homology. Yeah, I'll just go through this, run through this one, please. So this is a phylogenetic tree of 16S ribosomal RNA analysis of bifidobacteria showing the sequence homology with the database. So what we concluded out of this, that since in case of the pathogenesis of the ulcerative colitis, inflammation is the central event, and what we found in our study is there is elevated levels of bacteroids which can exaggerate the mucosal antibody, and because it also, for example, Bacteroides fragilis is known to generate BFT or the enterotoxins, and in case of decreased level of butyrate producer was supported by the SCFA concentration or short chain fatty acid concentration, which came down. There was an increased level of sulfate reducing bacteria that is producing more of H2S, which is toxic to colonocytes and inhibits the butyrate utilization. And there was an increase in the lactobacillus, as I told you, and we found an increase in lactic acid production. There is a loss of, which can cause a loss of mucin layer, because we found a decreased expression of one of the mucin gene. So perhaps the adherence property of lactobacillus is lost because of the loss of the mucosal layer, and therefore it transits to the feces and gives us a high value of lactobacillus. This is our conclusion right now. So what we finally say that the dysbiosis can be functionally coordinated, because the dysbiosis in IBD are correspondingly not simply the structural changes in the gut microbiota, but are instead associated with major impairments in many fundamental microbial metabolic functions with potential impact on the host. So there is a reduction in all these clades may thus have a functional consequence on the ability of the host to repair the epithelium and to regulate the inflammation. So this is my research team, and there is a poster presented by Rina who did this work, which is displayed on the poster session. Thank you very much.
Thank you, Dr. Paul. Would you like to just sit here? We'll take questions later.
Okay.
And our apologies, I think the introduction was a little inadequate. So Dr. Paul is a professor, School of Life Sciences, JNU, and also a UGC research scientist. We'll go on to the third speaker, Dr. Johan van Likamama Vliet. My apologies for the pronunciation. He's an R&D director, gut microbiology and probiotics at the Danone Nutricia Research. So welcome. We'll be talking on impact of food ingested microbes on commensal communities and host health. Welcome.
So thank you very much for this nice introduction. Congratulations with the pronunciation of my name. I think it was very good, actually. So. I want also to thank the organizing committee and the highly established scientific community here that organizes this meeting for inviting me. I am 2 meters and 4 tall, but I feel in fact very small in this order. So. You cannot hear me. Is it better now? I think I hear myself here. So what I will talk about is a sort of elaboration of some of the things that have already been introduced by others. And when I looked at the program, I tried to find some complementary entries to that. And what I will talk about basically is 3 things. First of all, I will elaborate a little bit more about where do these probiotic or food-ingested microbes, where do they come from? How did they end up in dairy, and how did they impact on our, let's say, our food production? And then, how do these organisms impact our gut microbial communities? And in the end, I would like to come to a sort of general model which maybe can guide also future discussions on where fermented foods and probiotics can have an impact in maintaining health and establishing health. And the first thing I really want to stress is that working for a food company, we are really about prevention and keeping people healthy. It's not so much about targeting diseases. And that's really, I think, the objective of the pharma industries. But we are really at this left side of the curve. This is a curve that I took from quite an influential document that really shaped also, let's say, the European Union research politics about a decade ago. Now, let's go to food-ingested microbes. I will sometimes use the word food-ingested microbes, sometimes probiotics, because I think probiotics has also in legal terms a very heavy meaning. It needs to be linked to demonstrated health benefits. I think it's broader than that. So you will see the 2 terms appearing. And the general hypothesis for a long time was that, in fact, these microbes that we use to ferment dairy products, they originate from, grass, for instance, plant material. And there has been a moment that either via a cow, the milk production machinery of a cow, or maybe directly, they ended up into dairy, and then they evolved and optimized to fermenting dairy products or even other food products. And then what happens today is, in fact, by consuming such products, we continuously challenge our microbiota. And then we have, of course, different aspects of the microbiota. We have the upper GI tract, where these ingested microbes are temporarily overwhelming really the resident microbiota. And then in the colon, they are still an important subdominant population. Now let's have a look. I found this paper in it's a Nature publication of a couple of years ago, and it's an archaeological study, and they found pottery in the Eurasia region. And you look at the quote that I highlighted here. It says that the first evidence of let's say processed milk, and in this case it means fermented milk, dates back to 6,500 years before Christ. So that's when dairying started. And then if we look at another, that's very quick. I will just continue. I think it's a mistake. So I looked at what does the word domesticate mean. Yeah? And if you look at the Webster's dictionary, it means to adapt an animal or plant to live in intimate association with or to the advantage of human. I think we can talk about our lactic acid bacteria actually as domesticated microbes, because this is what we do. So then, key questions can be, how was this human-LAB partnership established? What were, that's the scientist's view, what were the molecular events of domestication, if you want, and can we reproduce that in the laboratory? And how can we expand our partnership and exploit lactic acid bacteria better to our benefit? And it's a study that I started before I joined Danone, actually, but it continued during my period at Danone. We worked on Lactococcus lactis, which is a major component of dairy starters and of the typical Dutch cheeses, and it has two important main natural niches. One is, my time is up again. I don't know which button to push. Okay, no problem. It has two important main niches, that's dairy and plant material. And what we did is we had genomes available of two phylogenetically closely related strains, but isolated from these different niches. And that's the KF147. You will see the strain code a couple of times, and the typical dairy isolate is IL1403. So what a PhD student, a very talented PhD student in my lab at that time did, his name was Herwig Bachmann. He in fact did a laboratory evolution experiment, and that's very simple. So what you do is you put the plant strain in milk and you let it grow in successive re-inoculations and see whether a better evolved strain appears. In fact, there was one trick needed. You also needed to introduce a plasmid, which is required for growth. And then he went on. So in the end, he did that for 1,000 generations, which took about 5 months. So there was a lot of patience involved as well. And then he isolated from each of these 3 independent experiments single colonies and did growth and acidification tests and looked at fitness. And so how well is a strain able to compete with another strain? And we did genome analysis, so resequencing and also transcriptome profiling, looking at gene activation patterns. So what were the results? Well, this is what you see if you look at the typical experiment. You see the KF147, which is the highest curve, and then after 300 generations it goes quicker, and after 600 even quicker, and after 1,000 generations you really have quick acidification. This is an acidification curve. We found something strange, which I will not elaborate on. If you have questions, I could really do that after the talk if needed. But we found that we lost, in fact, the protease, and that is explained in a separate publication which was published in ISME Journal 3 years ago. And then the single colony isolates, they more or less reproduce this. So we have the full curve, which is the original strain, the wild type. Then we had one strain which had some sort of intermediate acidification acceleration, and 2 that were really much quicker. So what we did then is we resequenced these genomes of these single isolates. And then what do you find? Well, you find a whole bunch of mutations, and I only present here the SNPs, and what you see is that these 4 individual strains, they had between 3 individual mutants, they had between 4 and 21 mutations per clone. And interestingly, some of them were the same. So that really shows that independently you get the same mutation. That must be really a contributor to fitness. Now this is a very complicated slide, and I don't want you to understand it fully, I just want you to get the main message. So this is transcriptome profiles on milk. And the black dotted spot in the middle is the KF147, so the original strain on itself. Every gene you have the transcriptome mapped on itself. Then the IL594 is the dairy isolate. And what you see here is a straight line, and what you need to keep in mind is that the closer you are to the straight line of the next strains, the more close you are to a dairy-type expression pattern. And what did we see then? Well, if you put the original strain with the protease, you are very far off, because you're not in quadrant 2 or 3. And also this slowly growing strain is quite far off. But the 2 fastest growing strains, in fact, they seem to sort of tweak towards the dairy reference line, let's say it like that. We went further and looked at what type of things had changed in these transcriptome profiles. And what you see is that the adapted strains, they typically take up peptides, which is logical, because in milk you need to take up peptides to grow faster. And there was also something on branched-chain amino acids biosynthesis. Well, typical sugar uptake, other than lactose, was downregulated. So it really made sense in the sort of the ecological context. And we could trace that back to really the individual mutations. I will not show that, it's in the paper, but you can see that the mutations can really explain these differences in transcription patterns. So let's go back to this picture here, and now focus on the right panel of the slide. So how do these fermenting microbes challenge the gut microbiota? And what I will just want to do is to highlight something. We have already heard a lot about the gut microbiota, but I think we need to realize that from a sort of niche science area, it really has moved towards, I would say, a global and very, very important science domain. And for instance, the journal The Economist published on the cover in 2012 this as a main subject, and there's an extensive feature in this magazine. And also Science, the well-known scientific journal, chose it as the #1 scientific breakthrough area of 2013. So what do we really know? Because we see a lot of correlations, and here there's two slides, two pictures from two individual papers in Nature. And the left one depicts differences in the microbiome of obese and lean people. And I will not discuss it in detail, but I think what is interesting is that there are ecological models appearing. So we see that, for instance, short-chain fatty acid producers are down in obese people. We see that Enterobacteriaceae and Proteobacteria are going up. And we also see that global richness, so the diversity in the microbiome is down in some of these obese patients. And then the second study did a dietary intervention, an energy-restricted high-protein diet. And what this study found is that, in fact, some people are less prone to respond to this diet. And that is related to the microbiota composition. And if people respond well, you can increase the richness, the diversity in the microbiota. So that's very interesting, but still it helps to develop mechanistic models and testable hypotheses. I think it's very important for our science field. It also shows that diet is key. But we need to demonstrate causality. I think it's really, really important. Correlations only cannot be enough to tell the whole story. And this slide is a one-slide summary of a paper that we were involved in, which was published in Gastroenterology 2 years ago. And it's in fact a microbiota transplantation, a fecal transplant in people with metabolic syndrome. And it was consisted of 18 people, 18 obese volunteers with metabolic syndrome, and 9, so their colon was washed, and 9 of them, they received an obese microbiota. In fact, it was their own for ethical reasons, and 9 others received the microbiota of a lean donor. And the amazing finding was that, in fact, insulin sensitivity was largely restored in people that received the microbiota from a lean donor, showing really a causal component in the microbiota composition. And the microbiota analysis pointed again to this group of butyrate producers which we already heard about. So how can we really leverage this? I think I cannot stress enough that food is the main modulator of the microbiota. It was also already highlighted before. And then we had to talk about macroconstituents, microconstituents, prebiotics, and these food-ingested microbes. And of course, it's all in the context of epigenetics and lifestyle. And the last bit of my talk, I will talk about 2 specific models where we investigated this interaction further. So these models are, what we like to use is typically simplified models. That means healthy twin pairs, for instance, to eliminate some of the genetic diversity by human hosts, or dysbiosis conditions, typically in animal models. And the analysis is very much dependent on next-generation sequencing technologies. So 2 examples, an impact of fermented milk product in healthy twins, which was published in 2011, and one in a collaboration with Harvard Medical School, a truck model of ulcerative colitis. So both studies, they used the same fermented milk product, which consists of 4 strains, in this specific case, Bifidobacterium, Lactococcus lactis, Lactobacillus delbrueckii, and Streptococcus thermophilus. So this is the first study. It's a healthy twin pair intervention. It was done in the laboratory of Jeff Gordon. And basically what these twin pairs had, they consumed for 7 weeks the product, and then there were a whole set of analysis done on these on fecal samples, which I will not elaborate too much on. But one of the key findings was that if you look at the fecal microbiota, there's no major perturbation due to the ingestion of these food-ingested microbes, which makes sense because they are, of course, relatively low numbers. So the composition was not significantly changed. Then, if you look at one of the strains that was ingested, this Bifidobacterium strain, you see that if you try to do recovery from feces, you see that in this point of transient integration is nicely made. You see that only during the consumption of the product you can really recover the strain at significant amounts in fecal material. But there was something else which really surprised us. We also did transcriptome analysis on fecal material. And in fact what we found is that we could not only found a lot of transcripts of the bifidobacterium, which was highly active, but we also saw that just the presence of this strain in both the human studies and in the parallel mouse model, we found that carbohydrate metabolism was really significantly regulated. And there are 3 examples here of individual enzymes in plant-derived carbohydrate metabolism which were differentially expressed and activated in this specific case, both in a mouse model and in a human model. And it's extensively explained in this McNulty et al. Paper. The last piece of information comes from a model of ulcerative colitis. A TRUC model which is developed by Wendy Garrett and Laurie Glimcher at Harvard Medical School. And it's a genetic model, and it's characterized by a heavy, spontaneous, and a highly penetrant colitis, which can be treated by antibiotics. So by wiping out certain microbes, you can really treat or cure the disease, and it's transmissible. So if you co-house animals, TRUC animals with healthy animals, the healthy animals will also develop the colitis phenotype. And what the group did before we started to collaborate with them is that they nicely identified that certain proteobacteria are in fact part of the cause. So you can isolate especially Klebsiella pneumonia and Proteus mirabilis from colitis-genic animals. And then if you, in healthy animals, you put back these microbes, they will develop the TRUC. If you put these microbes alone, it's not sufficient for developing colitis. So they are really in concert with the endogenous microbiota causal agents. And then what we also did in a collaboration with them, we found, in fact, when we did 16S-based microbiota profiling, we found that one of the differences between the colitis-genic mice and the non-colitis-genic is that there's different levels of bifidobacteria. So upon colitis, you lose a large part of the bifidobacterial population, and that was confirmed by various methods. And then what we did is we did an intervention study with this fermented milk product, and we found that it ameliorates colitis. And what you see here is on the left, so you see here colitis scores. On the left you see the fermented milk product, then you see a non-fermented control, which is in nutritional composition the same, and then you see also a sham feeding control. You see that especially the fermented product has a very good performance here. And the other one shows, the next slide is a similar type of slide where we compare, in fact, irradiated product and product with live bacteria, and again you see that live bacteria do a much better job. Then we looked, as in the previous presentation, in fact, to what might be causal agents. And we hunted down really metabolites in the cecum. And one of the surprising findings, in fact, was that if you had fermented milk product, you had lower levels of lactate than in the control and in sham feeding experiments. This was sort of counterintuitive because we introduced lactic acid bacteria, so we expected more lactate. And a similar profile also for pH. And then when we looked further also to other short-chain fatty acids, we came to, we found in fact that though lactate was lower, so the other short-chain fatty acids, propionic acid, butyric acid, and acetic acid, in fact were increased upon the treatment with the fermented milk product. And we could link that back to the higher presence of specific butyrate-producing species. And then with subsequent the in vitro experiments, we found that short-chain fatty acid and pH, which was modified, really created unfavorable conditions for these colitis-genic microbes, Klebsiella pneumonia and Proteus mirabilis in vitro. And the last slide that I will show on research data comes from a publication which just came out a week ago, and it's something about, I wanted to make the point, contrast food versus medication. So what this laboratory did, they had various methods to treat the colitis phenotype in these mice. And the fermented milk product is only one of them, but also anti-TNF-alpha or antibiotic administration have the same effect. And if you then transplant the microbiota from cured mice to germ-free mice, in fact, the key message is that with the gentamicin and anti-TNF-alpha mice, they are really cured permanently. So the microbiota has been changed permanently, and it can this cured phenotype can be transferred. While for the fermented milk product, this is not the case. It's transient, and that is reflected by the presence of of the food ingested microbes in the feces of both the donor and the recipient. So here's my last slide. I would like to share with you a sort of general model of gut homeostasis and the prevention of disease, and where food interventions could play a role. And it's inspired on a picture, a figure published by De Vos and Newdorp in Nature last year. I think we can be in this homeostatic situation, and then due to infection or antibiotics or maybe diet or lifestyle, we slowly enter into a dysbiotic state. We lose some of the good microbes, if you want, and some of the bad ones, they go up. We maybe have impaired gut barrier function, which is very often seen in early dysbiosis, and there's always this component of low-grade inflammation. And if you don't do anything, it will go on and we'll get tissue destruction in the end, and that will lead to disease and maybe even chronic disease. So what you can do is, of course, you can go in with pharma, and you can maybe do gut microbiota transfer, and then try to reverse this process and ultimately get back to homeostasis. But I think where food should play a role is in this early stage, when things are about to go wrong. Then we come in with our functional or nutritional foods or specific probiotics to sort of close the loop and make sure that we go back again. And I think that should be one of our models, and this also helps us to think about functionalities that we want to identify in our probiotics, like restoring barrier function or maybe anti-inflammatory, which we can select and develop further. And this is the last slide with the key conclusions. I told you something about domestication. I want to make the point that these food microbes, they transiently integrate and impact the commensal structure in some extent, but especially transcriptional activity and metabolite production, at least when we look at feces. And I think there's really a need for further translational approaches to fully harvest the microbiome lever in food and health. And the last slide is in fact the most important one, because these are the people that did the work. I thank you for your attention, and I hope it was not too long.
Thank you very much, Dr. Blake. Would you please take a seat?
Yes.
So we come to the end of the session. Essentially, we had three speakers. Dr. Balamurugan told us how the microbiota changed during life, and he suggested that all these changes were attributable to changes in the diet, which modulate them. Dr. Paul talked about changes in inflammatory bowel disease and showed that active disease was associated with changes in the microbiota, which reverted to normal or reverted towards the baseline when they recovered. And Dr. Lee actually essentially told us that the gut microbiota could be modulated by externally ingested microbiota, and that would change both the microbiome and the metabolic capacity. And it had, and it's really the host and the microbe both acting on each other's genes to induce something. So if we have time for two questions from the audience. Dr. Ganguly, please.
My question is that Balamurugan shows that change of microbiota with age, and you showed a crosstalk. Do we have data that because some of the work which is coming up, that it helps in aging process, healthy aging process, the microbiomes. And so I would like to know from your basic work, both of you, what do you come across data set which shows that microbiota goes through some kind of a transformation during aging?
Is the question addressed to me, or? I can maybe give a start. So I think there are quite a couple of publications. I'm sure Colin Hill is... Does it work? Yes. I think there's quite some work going on that shows that in, let's say, in aging people, the microbiota is changed, and that correlates to immunosenescence or other adverse parameters. I'm quite sure that Colin Hill will talk about that tomorrow, because a lot of that work is going on in Cork, Ireland, actually. And yes, some of the signatures of dysbiosis, which I showed, low-grade inflammation, loss of certain beneficial bacteria like butyrate producers, is also seen there. So there might be, as with other benefits, good opportunities for probiotic or specific foods.
The only consideration is that there is this decrease in diversity in elderly population that we have observed. So any intervention that is possibly there to increase or even to elevate diversity would help.
One last question, Dr. Malik.
Yeah, my question is to Professor Paul. There is a confusion regarding the ratio of the formicutes to bacteroides. In literature, we find that in certain cases it is said that when the bacteroides are more, that is the ratio is less, it is better for the gut health. Whereas in others, we find that the number of formicutes is more and the bacteroides is less, that is better. So we are a bit confused in that regard. What is correct statement?
Hello.
Thank you.
Now, the thing is that, are you talking about the disease individuals or you are talking about the normal individuals?
Related to obesity.
My study is restricted to inflammatory bowel disease, and where we have seen that there is a drop in the formicutes and there is an increased production of the bacteroids. So the ratio actually changes.
Yeah.
Okay. And of course, I mean, this is only at the genus level I am saying. In the species level, that's, we can actually figure out. That's what I am trying to do now, to figure out in the species level, in the formicutes as well as in the bacteroids, if we can actually pinpoint which is the one which is going down and which is the one which is going up. But the ratio changes during the disease condition, not during the healthy condition. But regarding the obesity, I cannot make a statement right now.
Well, if you... I think many of us would be knowing about the papers from Kenny and Kenny et al. And the Dilzane. There, and if you read all those reviews, it's given that if the formicutes are...
Excuse me, Dr. Malik, I think we are talking about two different things. You're asking about obesity, and Dr. Paul's worked on inflammatory bowel disease. So I think we need to take this discussion outside during tea.
Okay, thank you.
I think we'll close the session. I'll hand this over to Dr. Neja.
Thank you, Professor Ramakrishna, Professor Shinchini Bhatnagar. Can we request you please to give a small token of appreciation? 2 minutes, quickly, to the speakers. And we can ask Dr. Sarah Thompson and Mr. Richard Hall to please come and felicitate our chairpersons. Thank you so much. This was a wonderful session, but we are running a little late, and because of a paucity of time, can I request you to please be back here after tea and a group photograph by 4:45. We are still late, even after we come here at 4:45. So we'll try to... Thank you so much.
Ini kaya sih.